dear students, myself Bhavani Pentela, working as an assistant professor in Noida Institute of Engineering and Technology, Pharmacy Institute, Greater Noida. In today's lecture, I am going to discuss about the signal transducer pathways and ion channel. So, without any further delay, we will dive into the session. So, in my previous lecture, I had discussed about the receptor theories, I had discussed about the classification, regulation of an receptors, drug receptor interaction. In today's session, we are going to discuss about the signal transducer pathways and about the one type of an receptor which is an ion channel receptor. In today's session, we are going to cover the ion channel receptor. Before covering the ion channel receptor, let me see what is a transducer pathway. So, what is a transducer pathway? What is a signal transduction? When you can call the particulars, how the drug will shows the signal. So, sometimes the signal transduction you can call as a cell signaling. The signal transduction is also called as a cell signaling. Here the drug when will enter into the body and it will bind to its receptor, it will convey the signal to produces some biological response, then you can call it as a transducer pathway. So, the signal molecule here the transduction, here the signal transduction is in a process where signal molecule that interact with the receptor inside or outside of a cell. So, what here the drug is doing here? Here the drug, what the drug in sense the signal molecule. So, which will carrying the signal? The drug, the ligand will carry the signal, right. Here when the signal molecule, when the chemical molecule enter in this process, if I am talking about the signal transduction mechanism, in this process what is going to happen? The signal molecule will passes the signal when it binds to the receptor which is present on or inside the cell. Most of the molecules that leads the signal transduction are the chemical substances. Whenever the signal transduction has been formed by the chemical substances like neurotransmitters, hormones, growth factors, all these are the chemical substances which will bind to its receptor to produce the signal transduction. And the signal transduction is very important for the growing of a cell or to work cell normally. If there is any abnormality of this cell signaling, which sometimes causes the cell death also, which can causes the cell which can transfer the cell, which can cell become like the cancer cell. Okay. And here what are the components of any signal transduction? So, here there are 5 components in the signal transduction pathway. First one is signal molecule. So, we need the signal molecule. Second one is signal receptor. So, signal molecule will bind to the signal receptor, and third one is signal conveyor. There is some conveyor which is present on the cell which will convey the signal when the drug bind to its receptor. And fourth one is signal transducer, and fifth one is transcription factor. Once the drug bind to its receptor, the signal will be conveyed by the signal transducer and it will shows the transcription factors. These are the components in the signal transducer pathway. If you see here, for example, agonist drug. So, we will discuss about here 4 types of signal transducer pathways are there: GPCR, ion channel, enzyme linked receptor and nuclear receptor, right. So, this is an agonist drug for an ion channel receptor agonist drug. So, imagine this is an acetylcholine drug. So, when the acetylcholine bind to its receptor, this is a signal molecule. Again, what is this? This acetylcholine is a signal molecule, and what it is? It is a signal receptor. When this acetylcholine bind to its receptor, then the signal will be carried. Which will carry the signal? Here the ions, when the agonist drug bind to its receptor, then the channel is going to open. Which type of channel is going to open? Sodium channel is going to open, and the sodium will enter into the cell and will causes the depolarization of an cell. So, this is one signal transduction mechanism, right. And second one is agonist drug. Imagine the same drug is an acetylcholine, okay. This is an acetylcholine or adrenaline, whatever it may be, right. So, when the acetylcholine bind to its receptor, this is a signal molecule, this is a signal receptor. When the signal molecule bind to its signal receptor, there is somewhere in that particular membrane there will be somewhere you can see the signal conveyor. So, for example, here the signal conveyor was an enzyme. Okay, once the drug bind to its receptor, here the enzyme will get activated and which will causes the cellular changes. So, the example was muscarinic acetylcholine receptor, because acetylcholine when it binds to the muscarinic, it will act to the GPCR receptor. And third one was when the agonist bind to the receptor, For example, steroids, if I am talking about the steroids, it is an intracellular receptor, it means it is a nuclear receptor. Where the receptors are present for the steroids receptors are present inside the nucleus, and there will be some changes in the intracellular prediction. You can see the transcription factors has been produced. And last one was enzyme linked receptor. When the agonist bind to this enzyme linked receptor, here this enzyme linked receptor will convey the signal, and this here the enzyme linked receptor, for example tyrosine kinase, it will convey the signal and it will activate the further process in the transcription factor. And if you see here diagram, you can understand in a very much better manner. So, this is a cell. Okay? This is first I had said you we are having totally 5 steps here. First one is signal molecule. So, this one is a signal molecule. What is a signal molecule? You can term as a ligand. It is either it is a neurotransmitter, either it is a hormone or drug or any substances or any chemical substances can termed as a signal molecule. This is a receptor. The signal molecule will go and bind to the receptor, and there is some signal conveyor which is present on some other part of a cell. So, where the signal conveyor will be present, it will present somewhere in the membrane. Once the drug bind to its receptor, the signal conveyor will convey the signal, and next one is transduction. Signal transduction. This is the signal transduction which will be happen inside the cell. The signal transduction process will be seen inside the cell, and which will activate the transcription factor, and this activation of transcription factor which can in turn which will happen inside the nucleus, the DNA will be converted into RNA, and finally protein will be get synthesized. If the protein is synthesized, then cell can causes the growth, and if the protein has not been synthesized, if there is no signal molecule, if there is no receptor, or if there is no transduction pathway, the cell may dies, which can leads to the cancer cell also. Sometimes the cell which can leads to the cancer cell also. So, in this signal transduction mechanism, we had discussed about the signal molecule, signal receptor, signal conveyor, signal transduction, and which will be happen inside the cell, and after that the signal transcription factor will be get activated, which will be converted into which we can see the DNA conversion to RNA, and finally the protein will be get synthesized. Okay. So, the signal transduction mechanism here, it has been classified, or it has been the signal transduction having the types. What are the 3 types of signal transduction? One is autocrine, second one is paracrine, and third one is endocrine. So, these are the types of the signal transduction. First one is autocrine. So, what is an autocrine? This is a cell, right? This is a cell. Here the cell will release the chemical substances inside the cell. Here the cell will release some chemical substances, and these chemical substances will go and bind to the same cell of the receptor. Then you can call as an autocrine. So, when you can term as an autocrine, here the cell will release some chemical substances or chemical messengers that will activate the receptor of a same cell. So, once the chemical messengers has been released from the cell, and this chemical messenger will activate the receptors which are present on the same cell, then you can call as an autocrine. And what is a paracrine? Paracrine, here the cell will release the chemical messengers, and these chemical messengers will activate the nearby cells or adjacent cells of a particular cell of for example, if you see here, this is this is a cell, right. So, here the chemical messengers has been released. So, the chemical messengers has been released from this particular cell, and this released chemical messengers will activate the nearby cell of its receptor. Then you can call as a paracrine. So, what is a paracrine? The cell release the chemical substances that will activate the cell surface receptor nearby its cell, right. And what is an endocrine? And what is an endocrine? Endocrine, it represents endocrine. The cell release the chemical messengers. Here also the cell will release the chemical substances, and which will be transported by the blood, and it will reach to the target site, and it will shows its action. Then it is called as an endocrine type, right. So, that signal transduction has been classified, or has having the 3 types of signal transduction. One is autocrine, second one is paracrine, and third one is endocrine. Autocrine, here the cell will release the chemical substances which will activate the same cell receptor. Next one, paracrine. Paracrine, by the paracrine, what you can say the paracrine, here the cell will release the chemical messengers which will activate then nearby the cell receptors, and third one is endocrine. Here the cells, what is going to happen here, the chemical messengers has been released from the cell and it will be traveled or it will be transported by the blood, and it will enter to the target site and it will shows its action, and it will enter the target site of a receptor and it will go and bind there, then it is called as an endocrine. So, see here, this is a cell and these chemical messengers has been carried by the blood and it had reached to the target cell and it bind to its the target receptor, right. And next one, signal transduction mechanism. We are having 4 types of receptors. One is gated ion channel, GPCR receptor, enzyme linked receptor and nuclear receptor, which you can call as an intracellular receptor. So, the transduction mechanism of each receptor will be differ, right. So, in gated channel, what is going to happen here, the transduction mechanism was the opening and closing of a channel when the drug bind to its receptor depending upon the ligand or the voltage of a channel. And next one, GPCR receptor, here when the drug bind to its receptor, then the GDP will be replaced by the GTP. By once the GDP is replaced by the GTP, here the G protein will be get activated, alpha subunit will be get dissociated from the beta and gamma, and in turn will activate the adenyl cyclase and it will shows the cellular actions. And what is an enzyme linked receptor? Enzyme linked receptor having the intrinsic activity, intrinsic enzymatic activity. And what about the intracellular receptor? Intracellular receptors are the receptors which are present on the present inside the cytoplasm or present inside the nucleus, and it will activate of this receptor result in gene transcription. Once the intracellular receptor has been activated, what is going to happen? Gene transcription you can see. So, these are the 4 signal transduction mechanism of a 4 receptors. So, what are the types of any receptors? So, here we having the 4 types of any receptors, type 1, type 2, type 3 and type 4. The type 1 receptor you can call as an ion channel receptor, type 2 receptor you can call as an GPCR receptor which is named as G protein coupled receptor, type 3 receptor you can call as an enzyme linked receptor, and type 4 you can call as a nuclear receptor or intracellular receptor. So, in this today's session, in this today lecture, we are going to discuss about the ion channel receptor. So, what is an ion channel receptor? So, here we are having the once the drug bind to this ion channel, the channel will be get open or closed depending upon the what the drug going and bind to its particular receptor. If I am talking about the acetylcholine, so acetylcholine nicotinic receptor, so when the acetylcholine bind to its nicotinic receptor, the channel is going to open. Which channel is going to open? Sodium channel is going to open. So, the acetylcholine is a cationic channel. So, it is opening the sodium channel. So, acetylcholine is a cationic channel. And what is the second drug? GABA. So, GABA A receptor is an ion channel receptor. Okay, it is an ion channel receptor. Which type of receptor it is? It is an anion channel. It is an anion channel and which will open the chloride channel, which will carries the negative charge chloride. So, it is an anion channel. And if I am talking about the glutamate, when the glutamate go and bind to the AMDA receptor or AMPA receptor, the opening of an channels like sodium and calcium will be seen, and this the glutamate receptors are the cation channels. And what about the glycine receptors? Glycine receptors are the anion channel. So, what type of channel it is going to open? What type of the channel is going to open by the particular drug or the neurotransmitter? That the channels are categorized into anionic channel or cationic channel. Here, if I am talking about the acetylcholine or glutamate, both are the cationic channels. It will open the sodium channel. And if I am talking about the GABA or if I am talking about the glycine, these are the anionic channels which are going to open the chloride channel. And here I am going to discuss about the classification of ion channel. Ion channel has been classified into 3 types. One is ligand gated ion channel, second one is mechanosensitive ion channel, and third one is voltage gated ion channel. These are the main 3 ion channels of an receptor. Okay, ion channel receptors has been classified into ligand gated, mechanosensitive ion channel, and third one is voltage gated ion channel. This ligand gated ion channel is further divided into 2 types: extracellular ligand gated ion channel, intracellular ligand gated ion channel. For example, if I am talking about extracellular, so here the example falls like glutamate, glycine, 5-HT3. So, these are the extracellular ligand gated ion channel. And what about the intracellular ligand gated channel? So, for example, G protein activated calcium channel, ATP sensitive potassium channel, this falls under intracellular ligand gated channel. And what about the voltage gated channel? Sodium voltage gated channel, sodium channel, potassium channel, calcium channel, right. So, these are the voltage gated channel. And depending upon the localization, the ion channel has been classified into two types. One is plasma membrane channel and second one is intracellular channel. So, depending upon the localization, which locality, depending upon the localization, the receptor has been classified into plasma membrane channel and second one is intracellular channel. So, plasma membrane channel example was voltage gated channel, sodium, potassium, chloride, right. And what about the intracellular channel? Sarcoplasmic reticulum channel, endoplasmic reticulum channel, myocardial channel, these are falls under the intracellular channels. So, here I want to explain you about what is an voltage gated channel, what is a mechanosensitivity channel and what is a ligand gated channel. So, first we will discuss about the ligand gated ion channel. So, what is a ligand gated ion channel? The name represents ligand. Here the ligand is going and bind to the channel. When the ligand binds to the particular channel, here the channel will going to open. So, the channel opening or close will depend upon the ligand. So, this is the reason why it is called as a ligand gated ion channel. Ligand gated ion channel, what is going to happen? Here the ligand will open or close the ion channel, right. It is not ion specific. The ligand gated channel is not ion specific. It is an if I am talking about this channel, it will open the gate. And what is a mechanosensitivity ion channel? Mechanosensitivity ion channel, it will open the gate and or it will close the gate in response to the physical force. If I am applying in some physical forces, by the physical forces you can open or you can close the channel, then you can call it as a mechanosensitive channel. In mechanosensitive channel, what is going to happen? In mechanosensitive channel, the gate will close or open depend on the force, depend on the physical force. And what is a voltage gated channel? Voltage gated channel is a transmembrane ion channel. So, if I am talking about the ligand gated, it is a multimeric ion channel, it is a multimeric protein, it is a multimeric transmembrane protein. So, voltage gated channel is a transmembrane ion channel. Here the voltage gated ion channel, the channel gate will be opened, which will depend on the difference of voltage across the membrane. If there is a difference of voltage across the membrane, according to that the channel gate will be get opened or closed. This is a ion specific. So, first one ligand gated is not an ion specific, voltage gated is an ion specific. And last one is a mechanosensitive ion channel. This mechanosensitive ion channel is due to because of physical force. Okay, and next one, here I am giving in some examples like nicotinic receptor, GABA receptor, serotonin receptor, glycine receptor, glutamate receptor. So, all these receptors having according to its action to its receptor, some receptors will show the excitatory action, some receptors will show the inhibitory action. So, if I am talking about the nicotinic receptor will causes the opening of sodium channel or potassium channel. So, what is next drug? GABA receptor. So, GABA receptor is an inhibitory transmission in the brain. It will open which type of channel it is going to open, what is the ion selectivity here? Chloride. And next one serotonic receptor. When the serotonic receptor when the serotonin bind to its serotonin receptor, it having the fast synaptic transmission and it will open the ions sodium and potassium. And if I am talking about the glycine receptor, when this glycine bind to its glycine receptor, it is an inhibitory neurotransmitter, it is an inhibitory, it will process the inhibitory transmission and it will going to open the chloride channel. And what about the glutamate? Glutamate is an excitatory neurotransmitter. It is a fast excitatory transmission in CNS, it going to open the sodium channel and calcium channel. And what about the P2X receptor? P2X receptor is a postsynaptic transmission which will sense the pain. Here it will open the sodium and calcium channel. So, if I am talking about these receptors, is a particular drug to the particular receptor, it will open the particular channel. So, the every drug cannot open the every channel or every drug cannot block the every channel. So, this is the thing, the particular drug, when the particular receptor has been gone and occupied, then it will causes the opening or closing of any channel. So, there is some connection between voltage gated ion channel and ligand gated ion channel. What is the relationship between, what is the similarities between the ligand gated ion channel and voltage gated ion channel? Both are the transmembrane protein molecules. So, if I am talking about the ligand gated ion channel, or if I am talking about the voltage gated channels, both are the channels transmembrane protein molecules. Both will activate the postsynaptic neutron. Both will falls under gated ion channel. Even voltage gated ion channel is also an ion channel, gated ion channel, or if I am talking about the ligand gated, it is also a gated ion channel. These are the similarities between the ligand gated and voltage gated channel. And if I am talking about the ligand gated ion channel, this is a cell surface receptor. The ligand gated ion channel, where the receptors has been present, the receptors are present on the cell surface. Here the drug will go and bind to the cell surface to its receptor. Once the drug go and bind to its receptor, it will open the channel. The opening of a channel will be seen in a milliseconds. So, the action will be shown very fastly once the drug bind to its receptor. Okay, so here I had given you some examples like nicotinic receptor. So, this is a nicotinic receptor. So, here the acetylcholine is a drug. Once the acetylcholine has been bind to the nicotinic receptor, there will be the opening of a channel. Once the channel has been opened, so what type of channel is going to open here? Acetylcholine. Acetylcholine is a cholinergic drug. It is a neurotransmitter, parasympathetic neurotransmitter. Once it bind to the nicotinic receptor, if I am talking about this acetylcholine having the two types of receptor, one is muscarinic and second one is nicotinic. Forget about the muscarinic receptor, because the muscarinic is a GPCR receptor. It won't function through the ion channel. So, but what is the receptor of nicotinic of acetylcholine? It is an ligand gated, it is an ion channel receptor. So, when the acetylcholine, so this is a drug, when the acetylcholine go and bind to the nicotinic receptor, then it will open the channel. Which type of channel it is going to open? It will open the sodium channel. Once the sodium channel has been opened, the sodium will enter into the cell and the cell causes the depolarization, the cell causes excitation. So, once acetylcholine come and bind to its nicotinic receptor, it will leads to the cell excitation, and which will be blocked by its nicotinic receptor blockers. For example, D-tuberculonin, succinylcholine. So, this will blocks the acetylcholine action on the nicotinic receptor. And what is the second receptor? GABA receptor. When the GABA come and bind to the GABA receptor, here the drug is a GABA, here the ligand is a GABA. When the GABA amino butyric acid when and come and bind to its receptor. For example, you can imagine this is a benzodiazepine drug. Okay, this is a benzodiazepine, or it is a barbiturate, whatever it may be. When this benzodiazepine drug go and bind to the GABA receptor, it is going to open the channel. Which type of channel it is going to open? It is going to open the chloride channel, because the GABA is an anion receptor, it is an anion type of receptor. So, it will open the chloride channel, the chlorine will enter into the cell, which will causes the hyperpolarization. Entry of a chloride channel, it will neutralize the excitation caused by the glutamate or any other excitatory neurotransmitter. So, it will compensate the excitatory action which produced by the excitatory neurotransmitter, and which will causes the hyperpolarization. So, these are the two examples which falls here. Acetylcholine is an anionic channel. Acetylcholine is a cationic channel, GABA is an anionic channel. When the acetylcholine bind to the nicotinic receptor, it will open the sodium channel. When the benzodiazepines go and bind to the GABA receptor, it is going to open the chloride channel. One will causes the depolarization, acetylcholine will causes the depolarization, and by opening of the chloride channel by the benzodiazepines will causes the hyperpolarization. Okay, next one. Next drug was glutamate. Okay, glutamate. So, if I am talking about this glutamate, students, here the ligand, what is the ligand here? What is the ligand here? The ligand is a glutamate. Here the ligand is a glutamate. So, if I am talking about this glutamate, here glutamate is a ligand gated ion channel receptor, it having the ion channel receptor. When this glutamate bind to its glutamate receptor, the channel will going to open, and these channels are the sodium channel and calcium channel. If I am talking about this AMPA receptor, because the glutamate having two types of receptor, one is AMDA receptor and second one is AMPA receptor. When this glutamate go and bind to the AMPA receptor, what channel is going to open? Sodium channel. When this glutamate go and bind to the NMDA receptor, then calcium channel is going to open. which will causes the depolarization of a cell. So, if you see here, the sodium channel will causes the depolarization, calcium channel will causes the depolarization. If the potassium channel has been opened, the potassium entering into the, if the potassium leaving the cell, especially in the myocardium cell, which will causes the repolarization, and opening of a channel of a chloride, is the chloride enter into the cell, which will causes the hyperpolarization. So, this is an ligand gated ion channel, students. Here we can discuss about the different types of neurotransmitter. So, if I am talking about these GABA and glycine, which will open the chloride channel, which will causes the hyperpolarization. If I am talking about the serotonin, glutamate or acetylcholine, which will causes the depolarization by opening of any channels. Thank you.